Among the blotting techniques, we studied southern blot and northern blotting technique, correct? The southern blot is for isolation of DNA and northern blot is for isolation and identification of RNA. Now, what is the usefulness of this southern blot and what is the usefulness of this northern blot? In one sentence, Sayon, in what purpose do we use southern blot? Some genetic disorders. In order to identify genetic disorder, number one. Number two, identification of person. Number two, identification of person. Like we have fingerprint, similarly we have DNA fingerprint also, okay? So by this we can identify person, we can identify genetic diseases, we can identify mutations, okay? So these three are the major applications of southern blot. What about northern blot? Okay, like almost half the student has come. So let me share. All your examinations will be preponed, which was supposed to be the university exam, which was supposed to be started from 13th February. Now it will commence from 27th January. So accordingly, all the examinations, whatever we have planned, it will be preponed. So better hurry up and buckle up. So by 21st, by next week, you will come to know the exact dates. Yes. So no time. No time. Life is full of surprises. Fine. So what are the applications of northern blot? Anyone? Application of northern blot? Some application of northern blot? Quickly. I have to teach the other things also. Stand up please. RNA? Anyone? Any applications you know? Follow? So what is the application of northern blot? Why do we do it? Separation and identification RNA. Yes. DNA? You go to DNA. DNA mapping. Northern blot, my dear. Northern blot I am talking about. It is used for isolation and identification of RNA. So what are the actual day-to-day life application? In many cases, we need to see, wherever we need to see the amount of expression of a gene. To actuate the amount of expression of a gene. Northern blot estimates the RNA pool, the amount of RNA present inside a cell of particular gene product. Okay? So expression, suppose some person is suffering from low hemoglobin. What is the cause? Which particular protein is being expressed less? How to know? But the gene mapping is fine, but still it is not being expressed properly. How do you know? The RNA population, if it's not normal, then the protein will not be synthesized properly. Correct? To know the gene expression, amount of gene expression. Okay. Now, translation has been done. Fine. So, we know about protein synthesis. Now, how to determine, how to find out or identify a protein, a DNA. Before I go, just remember, always remember these steps of these blotting techniques. Very, very useful. In examinations you will get, definitely you will get. These are the steps of southern blot. Northern blot steps. Remember the steps, nothing else. Now, the western blot is about identification of protein. About identification of protein. So, how do you identify a protein? Eat the protein, taste it and tell. Correct? But not all protein are so tasteful. Rather, can be distinguished by their taste or smell. Correct? Like milk protein, casein, can be distinguished by its smell. Albumin can be distinguished by its smell, egg albumin. Right? But not all protein. Hemoglobin cannot be identified by the smell or taste. Fine? So there are numerous proteins. So how do I identify it? Number one. This is why western blot. So in previous blotting techniques, what we have employed? We have employed one electrophoresis procedure, by which we separated the fragments of nucleic acids based on their size, not charge, based on their size. But problem with proteins are not only charged, but they also alter their charge with change in pH. The same protein at pH 6.5 will be negatively charged, at pH 8.8 will be positively charged. Correct? So we need to, whenever we ascertain a particular pH to a buffer, we also should know that what would be the charge of the protein of our interest which we want to separate. So in order to disengage the effect of charge on the protein and bring it down to its only molecular weight, so that we can separate the proteins by their molecular weight only, we have a procedure. But moreover, another aspect which is different from nucleic acid is protein has got different shapes. Like we know collagen, it's a long protein. But globin, hemoglobin, they are globular protein. So in a meshwork, which protein will move faster? The one which is long in shape, elongated, or the one which is globular in shape? Yes, the globular will move faster. So again, now we have disengaged the charge. Now we have disengaged the shape. Now protein with its natural shape and charges, when electrophores, it's called native gel electrophoresis, without harming the normal charge and shape of the protein. Native gel electrophoresis. Now, when we alter it, when we consider only the molecular weight, not the charge or the shape of the protein, we change the protein's shape to shapeless and charge to a given particular negative charge. All proteins will be negatively charged. Only difference in their in them will be their molecular weight. This is called denatured gel electrophoresis. So one is native gel electrophoresis, another is denatured gel electrophoresis. In order to perform denatured gel electrophoresis, we use certain detergents. detergents. Detergents are negatively charged. They will cling to positively charged protein molecules. So no protein will be positively charged. They will all be negatively charged because of detergent. Now which detergent we use? Can you name any detergent? Any detergent. What is the use of detergent? Have you used detergent in your so far life? Used. For what purpose? Now that is a science. I asked you, have you used detergent in your life or not? Yes. For what purpose? Washing clothes, hands, correct? So what is the compound? There, sodium lauryl sulfate, SL. Do you buy shampoo bottles? Just look out at the back. What is detergent you use? SLS. Correct? The SLS is used for washing our hair. It is so healthy that if it touches your skin for a long period, your skin will be gone. All lusters of your skin will be gone. It is so healthy. Right? It is used for denaturation of protein. It is so healthy. SDS is used for this purpose. Sodium dodecyl sulfate. SLS can also be used, but SDS is nominal, less costly than SLS. It can be used. Okay. So usually SDS is used for electrophoresis. Now these are the steps or flow chart. Electrophoresing the protein sample. Electrophoresing the protein sample. Now, suppose we want to identify hexokinase. Have you heard hexokinase? Huh? Where do you find hexokinase? Huh? Where? So, choice of the source depends on the extraction process or directs the extraction process of the protein. If it is bone, we need very hard machinery to crush it, then to dilute it, then to separate it using a buffer. It's a huge job. And moreover, you have to be very careful that it should not harm the protein itself. You must not wash your clothes so nicely that it gets torn. You should be able to be wear after washing. Otherwise, it's of no use. Fine? So that is a issue. The choice of the source and the process of extraction. Next, assembling the western blot sandwich. What is this sandwich? The one we had just now? What do you mean western blot sandwich? Is a protein, antibody. Suppose we take a protein A. We need to find antibody which is anti-A. Fine. Now we need another secondary antibody which will recognize anti-A antibody. So first then we have to assemble, we have to gather information regarding the protein and their antibody and their antibodies. Primary and secondary antibody. So what do you need? Protein, primary antibody, secondary antibody. If the protein is an antibody, then correct? Antibody is also antigen for some other animal, organism. So we have to grow antibody for that antibody. Fine? So it will still be antibody. When we are talking about this thing, the role of the first protein, which is to be isolated, is always antigen, whether it is antigen or antibody. Its name may be antigen or antibody, but we will consider it as antigen, and we will have to develop specific antibody against it. Fine. How do you develop specific antibody? There is a technique called hybridoma technique. We will learn it. Don't worry. Next, transferring protein from gel to nitrocellulose paper. How do you transfer it? How do you transfer the fragments of DNA from gel to nitrocellulose paper? Because yesterday only we had a class on stress busters. Fine. So Western blotting. So even if you tell something hard, something rude, you must talk it softly. Like, I'll be very pleased if I can slap you. Rather saying, I'll slap you. See, the effects will be much more soothing. Yeah? I had many experiences with my father like this. He used to call me, suppose I have done something mistake. So he used to call me, he never used to get angry, show anger from distance. He always used to call me, come buddy, with a smiling face. And okay, fine, I used to go like this, smiling face, and very soon my smile used to go away, and eyes would be full of tears. The effect, you know, what is the reason. So anyway, so we learned the stress busters many years back. So transferring protein from gel by blotting technique, it could be mechanical blotting, it could be electro-blotting. If a peristaltic pump is used, then it is called mechanical blotting. If electrophoresis means two electrodes are used for blotting, it is called electro-blotting. Staining of transferred proteins. Proteins cannot be visualized without staining. Why? The color of nitrocellulose membrane is, don't start calling her NM. It's nitrocellulose membrane, in short. Fine. I love blue color. My favorite color is blue color. Stress busters. Had it been green, I would have said green. Had it been yellow, I would have said yellow. You know? Although they are saying yellow, yellow, dirty fellow. You know? White, white. Huh? There was one chewing gum came, Orbit White. It was his promotional song. So transferation by staining. How do you stain? In histology, what do you use for staining tissue? Yes, so those kind of stains can be used. Fine. But here we are actually working with proteins which are still alive with their antibody activity. So how do you separate? If you want to keep the property of the enzyme or the protein intact, you need to separate by direct visualization method. So it could be fluorescent method, it could be radioimmunoassay method, or it could be just, no, not chromatography, just autoradiography, you know, adding radio label tagged antibodies and identifying by autoradiography, adding fluorescing substance to the antibody and identifying their position by visualization, by UV irradiation, or these are harmful, or using GFP, green fluorescence protein. What is GFP? GFP is something for which the discoverer got Nobel Prize after 25 or 27 years of the discovery. Means the Nobel Prize has a notion, unless it shows some effect on mankind or society, it would be given Nobel Prize. So it took 25 years to show its importance in our day-to-day life, in science. Then he was given, a Japanese person. He isolated GFP, green fluorescence protein, from a natural source. It is very harmless. It has no harmful effect on nature, on environment. So it was all the RIA technique, radio immunoassay techniques and everything, radioactive level proteins, they were soon replaced by GFP and more environmental friendly. You know? Actually, what happens in a laboratory after working with these radio immuno tagged substances, we accidentally, unknowingly, we used to throw it away with normal drainage system. You know? So if it goes to somewhere in the drainage system, it goes to sea, if it goes to some other parts of the water bodies, it might create havoc. You know, in an exaggerated form was shown in a movie called Godzilla, where one small lizard became a large Godzilla due to radioactive emission, mutation, by some atomic explosion caused by, the name of this nation was France actually. You know? So anyway, they tested a lot of many atomic bomb explosion in the Pacific Ocean, so which caused many radiation, and from there the line of the story came up. Fine. So in order to keep your environment healthy, the use of green fluorescence protein became more and more rampant. Fine. Now, blocking non-specific antibody sites to the nitrocellulose paper. What is this? The nitrocellulose membrane is like a sheet like this. So on a small portion will be occupied by the protein of our interest. Rest all portion will be like empty chair. So what might happen if you open the door? Anyone can come and sit, and we might misrecognize them as our student. Right? If I come new, fine. So these seats should be blocked. Now you are students, there is a gap inside, in between. Those gap has to be blocked by non-specific proteins, so that no stain is also chemical, it can cling to the seat without having any protein, so it will give a false reading. If a staining molecule go and sits in these vacant places, then I will think they are students. So in order to skip that misconception, they used to block those places by some non-specific proteins, because empty seats might attract those staining molecules. But if the seats are blocked with specific protein which are not to be stained by that particular staining agent, then they will not hold the stain. Am I clear to explain this? So the empty seats, if they are occupied by the non-specific proteins, they will not cling to the staining molecules. So there will be no false positive test. Fine? This is called blocking. Usually it is done by a sheep RBC protein, sheep albumin or bovine serum albumin, like that. Now probing the ectoported proteins with primary antibody, washing with non-specific antibody. If anything is extra is there, should be washed. Then detecting antibody by horseradish peroxidase. One is the enzyme. Initially this was a process they used to use, horseradish peroxidase. It is an enzyme which has got peroxidase activity. Now with which, when we add this substrate, they produce a green color. So that will show the first presence of protein on the nitrocellulose membrane. That our protein of interest is present on the nitrocellulose membrane. Right? And that too with all their normal natural properties. It is not wasted. Photographing the immunoblot. Take a photograph. Just a normal pinhole camera, take a photograph. Fine? So now we know that this is an electrophotogram, that when we separate this such and such protein, we get the band at this region. Fine? So whenever next time we will do it for actually isolation process, we have to maintain, we have to identify those regions only and cut those portion of the gel and collect the protein sample. Fine. Then we can use a protein sample for further amplification. Clear? This is for identification and isolation. Terms are identification and isolation. No, southern blot, identification and isolation of DNA. Northern blot, identification and isolation of RNA. Western blot, identification and isolation of protein. Clear? What are the major difference between these two? The nucleic acid bonding technique and protein bonding technique? The gel which is used. The nature of the gel used for electrophoresis is different. Huh? Here we are using SDS. If we use SDS for DNA, it is gone. RNA is gone. The nucleic acid is damaged. You cannot use it. We have to wash with detergent many times. If you wash it with detergent to nucleic acid, you have to remove the unwanted proteins, because DNA is also associated with many proteins. You have to remove those proteins. We have to use some protein eradicating substances like some very mild detergents, which does not affect the nucleic acid but remove the proteins only. Fine. So these are the processes. Now, what is another difference? Usually, nucleic acid is electrophoresed on horizontal electrophoretic chamber. Proteins are done usually on vertical electrophoretic chamber. This is a diagram of electrophoretic chamber. These small holes are called wells. They are made while casting the gel using a comb, churning, which leaves behind, after the casting of the gel, these gaps. One of the gap is used for, see the color of the solution, blue. Right? It's called, it is used for marking the extent of migration of the fluid molecule. We don't know how far the protein will go. If you run for 1.5 hours, the protein might cross the whole length of gel and go to the next buffer chamber. We might lose protein. So we leave a ladder sequence, a marker dye. It is blue Dextran blue. Dextran blue. It is a very high molecular weight, blue color solution, dye, Dextran blue, which is given here. So high molecular weight means its molecular weight is higher than the proteins of our interest. So even if it crosses the whole length of gel, it is unlikely that any of the protein molecule will cross. Right? So high molecular gel will do, and proteins will be here in between. And then one more well is used if we don't know exactly the size of the protein. In that case, we call ladder sequences. Ladder, L-A-D-D-E-R, means moi, jeta ব্যবহার করা হয়, তাই না, for climbing walls. Ladder sequences. So ladder looks like different steps. Now these steps means actually proteins of known molecular weight, so that whenever we identify our protein of choice, directly we can compare their molecular weight with the known molecular weight protein. Understood? So at the same time, in the single run, we can not only know their location but also know their molecular weight. If we apply a Dextran and followed by a ladder sequence, which contains proteins of different molecular weight, known molecular weight. Clear? For comparing the electrophotogram of our protein of interest or protein to be separated. Electrophotogram, remember. Next, so these are the proteins. See, they are two polypeptide chains. They are joined by disulfide linkage. So it should be heated or some chemical can be used. It can be 8 molar urea or mercaptoethanol. Both the reagents are used for denaturation of proteins. They separate the polypeptide chains from each other by dissolving disulfide linkages. So if it is asked which of the following compound is used for dissolving disulfide linkages, MCQ. It could be mercaptoethanol, it could be urea solution, 8 molar urea. Clear? Now this one, after separation, now it is loaded with, what is that? What is that? SDS, the detergent. See, all are negatively charged. Now whole protein is negatively charged. So what is only different in their molecular weight? Charge-wise there is no differentiation. Shape-wise there is no differentiation. Only thing that will differ in them is molecular weight. Now if we perform this electrophoresis, this particular electrophoresis is called SDS gel electrophoresis. Denatured electrophoresis. Without adding SDS and mercaptoethanol, it would be native gel electrophoresis. Clear? This diagram? Any question from here? Now, after transferring to nitrocellulose membrane, because in the gel it is very difficult to perform the further operations. So we transfer it to nitrocellulose membrane. Now this is protein blood and nitrocellulose membrane. Now they have made some marking here, but in actual life you cannot see those. Why? Light blue color. It is blue color, so you cannot identify any protein in that. Right? And nitrocellulose membrane is very brittle when it is dry. It should always be soaked in buffer prior to use. It's such a paper, it's paper. Just like your paper of your copy. Instead of using cellulose, here they have used nitrated cellulose. This is made up of cellulose, this is made up of nitrated cellulose. Only difference is this. But that makes this paper is very brittle, like paper, thin paper. You cannot hold it like this, it will break. But the moment you soak it in buffer, now it is very sturdy. Now if you do like this also, it will not break. So that membrane is used for transferring the protein. Now on that, you must use this thing. Specific antibody. This one, which recognizes that particular protein of interest. And this one should be recognized by secondary antibody, which is not shown here. To make it more simple, they made it like that. They directly added the enzyme here in the primary antibody. Fine. But in order to identify more precisely, we always use a sandwich method. Means antibody to that antibody, another anti-antibody. Okay, that gives us more precision. Fine. That identifies this is a location. So now if this is a location, that means in gel it will come here. So if we take a photograph and next day when we perform the gel electrophoresis for actual separation of the protein, only this portion of the gel we should cut and collect the protein. We can collect the protein from here, but it will not be sufficient for our other purposes. If we want more amount of protein, then we should take it from gel. And we can increase the amount of protein to be separated by increasing the thickness of the gel slab. In order to make only SDS page and identification of protein, say for example, identification of HIV virus genome proteins, we need not to increase the thickness of the gel because we have nothing to do with the known virus, known protein. What I will do with taking the viral protein from the patient body? It is of no use. Can I able to sell it? HIV le lo, पाव किलो 10 रुपए, पाव किलो 10 रुपए, है ना? Correct? So, for detection purposes, diagnosis purposes, the gel slab is made very thin, like 1 millimeter or less than that. Fine. But for other purposes, like for collection of protein, suppose you want to separate chlorophyll, particular type of chlorophyll, then the gel slab increases to up to 5 millimeter. That is very, very thick. Fine. It can collect to around more than a gram. See, this has shown this sandwich method. This is antigen, target protein. This is a primary antibody. Secondary antibody attached to it, one enzyme. In our previous description it was hostess peroxidase. Here it is alkaline phosphatase. So here it is used PNP, pan-nictophenol phosphate. PNPP will remove the inorganic phosphate from the PNPP, alkaline phosphatase, and the end product will be PNP, pan-nictophenol, which is yellow in color. So formation of yellow in color means the solution contains this enzyme. Solution contains this enzyme means the solution contains this antibody, and presence of this antibody means presence of this antibody, and presence of this antibody means presence of this antigen. Is it clear? So this is called sandwich method. Similar technique is used for ELISA. Same thing. But the instrumentation is different. In ELISA and in colorimeter, in both the cases, we estimate the density of the color by colorimetric method. The color should be in the visible range between 400 to 700 nanometer. Fine. What is the difference in estimation measuring the density of the color in colorimeter instrument and in ELISA? In colorimeter, you put one glass tube, cuvette, inside the hole of the machine, right? So there, light passes from side to side. The path length is 1 centimeter, fixed. The diameter of the tube is 1 centimeter, fixed. Be it cylindrical, be it square. The diameter will be, the light path length will be 1 centimeter fixed. In ELISA, the light passes from bottom to top. So even if you take in colorimetric estimation, if you take, suppose instead of 1 ml, if you take 2 ml, 3 ml, 5 ml sample, the value will be same. But in case of ELISA, since it measures from the bottom down to up, if the volume is different, the reading will be different. So pipetting error, ELISA is much more sensitive to pipetting error. Why? Because the way they measure, they throw the light from bottom to top of the well, small well. So if the total volume of the sample is 300 ml, it will have some reading. If the total volume is 350 ml by not ml, microliter, then it will be having different with same color but different value. Correct? Is a difference. Anyway, you might be knowing already. Okay. What is this? See, UV irradiation. Correct? Now this is a particularly, this is a chamber, like a box-like chamber, which is closed by lead coated metal sheets. Inside there is an UV light. Here, this is closed like this. Here one UV light is there. We place the slab here. We see through a very thick glass, because UV light is very harmful for our eyes. If you continuously view through UV light, what will happen? Early cataract. Early cataract. Fine. So we can see them glowing. How? How? If there are some fluorescent dyes present there, say for example, ethidium bromide. Which of the following is a fluorescent dye? But which of the following is not a fluorescent dye? Ethidium, actinium orange, riboflavin, calcitonin. Options are very easy to identify, correct? So by this, positively, stress busters, I gave you 3 answers which are fluorescent. So what are those? Ethidium bromide. See. Huh? Fine. This is an old way for blotting technique.
Okay, good morning everyone, and which is a broad topic. Now we'll narrow it down to discuss today the portions of the year, and if we have enough time permits, we'll talk about these little expansions of an year which is related to a part of year. Now today's learning, what we are going to do, we are going to precisely talk about the middle year. So we have the year which is split into 3 segments: inner year, middle year and external year. Now we restrict the discussion today on the middle year and the relationship features which is present in the middle year. Now in the middle year, we'll have the discussions which is related to this particular middle ear cavity. The middle ear cavity itself we call as tympanic cavity. So don't get fooled down in an examination if the exam question discuss about tympanic cavity. The tympanic cavity and the middle ear cavity are the same thing, which is reciprocal and indicating the same thing. So the tympanic cavity and the cavity of the middle ear is the same thing, same entity. Now today we will be precisely talking about this tympanic cavity or middle ear cavity. That means it's a cavity have got some contents in it. So we'll be going to talk about what are the structures that is present inside as a content, and along with that the boundaries which is related to it, and it has some kind of margins. We call them as walls. So this tympanic cavity have got walls, floors, roofs, and they have features in them. So we'll be talking about those features which are present in this middle ear. Now whenever we're going to talk about this middle ear or the cavity of the middle ear, there are 2 things we will be focusing here. One is, one of the cavity wall of the middle ear is formed by a membrane. We call them as the tympanic membrane, also commonly known as eardrum. So we'll be knowing this eardrum. It could be a separate isolated short note which may be involving this tympanic membrane, also known as eardrum. Or maybe it could be a part of a long portion fragmented, just like a generalized tympanic membrane or tympanic cavity together and split into small portions. So today we are going to talk about the tympanic membrane, we are going to talk about the contents of the middle ear cavity, and then we are going to talk about and discuss about the features. Now whenever you are going to talk about the features, we will be narrow down this essential or key features on the walls or this cavity has. And we should be able to write it down, reproduce it in examination if the question sets in during the examination about the cavity of the middle ear or the features which is present in the cavity of the middle ear. Alright, and along with that we will be talking about the clinical aspects, and again I repeat, so whatever you are writing in anatomy in the gross, your answer should include 1-liner or 2-liner related clinical anatomy. If you are failing to write that, you are losing the marks. You are just not answering any clinical thing. That means you are not getting the marks on it. No matter how you good write on the rest of the content, if you are not writing any single words about clinical thing, you are losing marks. So even if you have written each and every words in a book, whatever it is there, failing to write the clinical anatomy related to that part, you are going to lose some mark for sure. Alright, so this course or curriculum is involved on the clinical anatomy. So you have to write accordingly on this part. So let's begin with this. So we'll be precisely taking 20 minutes to talk about it, 20 minutes to talk about this middle ear cavity, and then we'll talk about the clinical aspects as well. So this is what we are going to talk here is the ear. And we know that this particular ear is split down for the descriptive purpose into three segments. External ear, which has the pinna and the tube connected to it, external acoustic meters, the yellow shadow. Then it lands into this particular blue domain, which is the point of discussion for the day, which is the middle ear, and this particular pink region, which is the deeper side of the ear, we call them as inner ear. We'll skipping this yellow part and the pink part, we'll be focusing on the middle part of the ear. But to introduce this, these are the things which is present in it. So whenever you're going to talk about the middle ear, the state of this discussion starts with discussing with the partition which is present in here between this external ear and the middle ear. So this between the external ear and the middle ear, we have a boundary which is existing in here. If you're just like throwing light through the external ear, it is reflecting on some structure which is partly white. That's the thing we call them as eardrum or tympanic membrane. So this is what we're starting our discussion today. Tympanic membrane, which I have mentioned, is a very important entity in a clinical aspects is concerned and as well as the anatomy part is concerned. So this is what you are seeing in a magnified version is a cross section you are seeing here. Ear ossicle external acoustic meters or auditory tube, external tube, which is having a two walls, anterior wall and the posterior wall, and fitted on the end of this particular tunnel is this particular radar-like structure. We call this particular radar-like structure as a tympanic membrane. So we have to know what is tympanic membrane. Now this particular tympanic membrane is a kind of partly white trilaminar membrane which is present in here. So it's having three layers. That's what you have to memorize or remember. It has three layers which is precisely we have to know. It's a trilaminar transparent partly white membrane which is placed inside this between this external ear and the middle ear. It's present on that part. Now this particular partly white membrane, what we were saying here, is having three layers. Is having this particular three layers which is present in here. We have to know precisely what are the three different layers which is present on it. So if you're going from outside to inside, it has a layer number 1, outside to in. You have an external layer, we call them as cuticular layer. That means it's covered with the skin. The second layer, deep to this, we have a fibrous layer. That is made of fibers, which is present in it. And the third layer, which is the inner side, which you cannot see no matter what, which is present inner side of this cavity, which is known as the mucus layer. So these are the three layers of this tympanic membrane has. And we have to know these three layers and where from these three layers are sourced embryologically as well we have to know. Now this particular three layers has a three different origins embryologically. So this outer layer, which is a cutaneous layer, which is developed from ectoderm layer. The middle one, the fibrous layer, is mesodermal in origin. So they are developed from the mesoderm layer of this developmental origin is concerned. And this mucous layer, which is innermost layer, which is developed from the endoderm. So we have three layer membrane existing in here, which have got three different layers, have got source from three embryologically different origins. Ectoderm, mesoderm and endoderm, three of them are present in here. Now, these entities you have to remember, this have to memorize. Now talking about this tympanic membrane, if you are grossly seeing this tympanic membrane, it looks like a semi-transparent structure which is present in here. Now this particular structure which you are seeing the margins of this, it has a peripheral margin which is lying on the margins over here, which is a peripheral, and this part is the central part of the membrane. Now this particular peripheral margin which you are seeing here having a fibrous ring. So the outer margin, the peripheral, has a fibrous ring. That fibrous ring is fitted into a bony socket. We call that bony socket as tympanic sulcus. So this outer margin has an extra layer which is known as fibrous ring kind of thing, which fitted into a bony socket, we call them as tympanic sulcus. Now this particular tympanic sulcus having these attachments and the 2 points on these regions makes this particular tympanic membrane split into 2 domains. It has 2 domains to look into it. A part of tympanic membrane, a very tight and tense structure, and a part of tympanic membrane have a very slacky part and a very loose part. So if you look at this particular tympanic membrane, you see this triangular or kind of like a piece of cone area which is lying over here on the top. So which is having a different color compared to this one and that one. So this is a little bit pearly white, this is like a pale cream. So this area which is a small narrow area which is lying on top, which is a little bit loose and slack, that part of the tympanic membrane is a flaccid. We call them as flaccid part of the tympanic membrane. Whereas the rest of the tympanic membrane, which is 97 to 99% of the tympanic membrane, which is tightly fitted, we call them as pars tensa. So whenever you are going to talk about the parts of this tympanic membrane, it has 2 parts, part A and part B, which is a major part which is tense. We call them as pars tensa part, and a minor part on the top or superior aspect, we call them as pars flaccida, which is a loose fitting structure right now which is present in here. So these are the 2 parts which is this tympanic membrane has. Now coming to the surfaces, it has 2 surfaces. One is external surface. If you throw the light through the external ear, you can see that part, which is a lateral surface or external surface. The part which is lying towards the inner side of the middle ear cavity, which is the inner aspect, which you cannot see, which is the medial side or internal surface of the tympanic membrane. So it has 2 membrane side, external side, which is known as lateral side. This has got other side which is present towards this middle ear cavity, we call them as internal side, also known as the middle ear side. Now, how this tympanic membrane is placed? The tympanic membrane is placed like a radar which is going to catch some signals. So if you are looking this particular tympanic membrane, it is not vertical structure, it is a little bit curved. Now this particular curvature which is present in here, it is the middle part of the tympanic membrane is curved towards the cavity of the middle ear. That means this tympanic membrane which is fitted like this. So if it is a cavity side towards this tympanic membrane, it is curved like this. So it has a bulge naturally towards the cavity of the tympanic membrane, just like looks like a disc antenna whichever we have this receiving the signal from. So having this particular thing placed like this, it can catch the sound amplifications which is better coming from the ground or coming from heights or coming from sides. So this particular tympanic membrane which is having a bulge or kind of like a cavity side projections towards this tympanic membrane. Now this particular bulging, as you are seeing here, which has a maximum point of bulging somewhere near in the middle part, that maximum bulging point of the tympanic membrane, we call them as UMBO, UMBO. Alright, so these are the general features of this tympanic membrane. So it has 2 surfaces, lateral surface, medial surface. It has 2 parts. One is tense part, other is flaccid loose part. This has got kind of 3 layers, sourced from 3 different sources development origin. And this particular tympanic membrane is not a flat structure, rather it is carved or bulged towards the cavity of the tympanic membrane, and the highest point of the bulging we call them as UMBO, umbo. Alright. Now, the other two aspects about the general features of the tympanic membrane. Now, if you seeing this particular tympanic membrane, we are seeing this particular layers, which is outer layer, middle layer, and this innermost layer. The fibrous layer, within this fibrous layer and this inner layer of the mucous membrane of the tympanic membrane is bedded here an ear ossicle. We have the middle layer fitted with the 3 ear ossicles, which are small piece of bones. So those ear ossicles, they are present in here. One of the ear ossicles are bedded within these layers of these tympanic membrane. We have a bone or ear ossicle, we call them as malleus. Now this particular malleus has a handle. That particular handle of the malleus is the fibrous layer and the mucous layer of this tympanic membrane. So this is other feature which is present to it. Now why this particular structure is fit into it? Whatever the sound amplifications are picked up by this particular tympanic membrane, that needs to be transmitted to the deeper side where it can be processed. So to pick up the sound from the tympanic membrane, we need a transporter. So one of these transporter which is picking out the sounds from it is a bone ear ossicle, which is a malleus, which is the part of the bone is grooved inside the layers of the tympanic membrane, and the part of the malleus which is fitted in here, we call them as malleus handle. Okay. Now following this particular malleus handle, it is crossed by a nerve which is not supplying to it. So the nerve which is crossing over this tympanic membrane to different directions, medial to lateral or whatever it is, we have a nerve going across, we call them as chorda tympani. So we have these particular features which is related to it. So this is the thing we have talked about, oval transparent thing which has the thin layers, 3 layers, outer side skin and inner side mucus, in between fibrous, sourced from different regions, maximum bulge point, we call them as UMBO. And it is sucked in, and as we are seeing here, the tympanic membrane has a bulge which is going inside, and this part we are talking here is that handle of the malleus which is going down and fitting with this particular fibrous layer and the innermost layer, the handle of malleus which is getting over here and ending on the point which is reaching towards the umbo region of this tympanic membrane. Alright. Now, along with this particular structure, this tympanic membrane is crossed by a nerve which is running across, which is known as chorda tympani. So, if you are seeing this particular picture, somewhere in this region you see a nerve, pink structure crossing over, which is a chorda tympani, which is a branch of facial nerve. We know the significance, or maybe we will be coming across the significance of it later on, because it is beyond our discussion topic. Today I am not going to discuss here, but you have to know this particular nerve is crossing over. This is a general gross feature about the tympanic membrane. So whenever you are addressing, these are the general features of the tympanic membrane or highlights of the thing that you have to mention in a schematic or whatever the way, whatever the matter, that includes this particular region of this clinical discussion, and as well as embryological source as well on this tympanic membrane, which is a pretty common and easy to memorize. 3 sources from 3 different elements. Now, coming from these particular parts of these things, pars tensa, pars flaccida, which is lying between this particular layer, majority is tense, and little bit part which is lying over here, pars flaccida, which is there. Now, coming to this particular nerve supply of this tympanic membrane, it has 2 surfaces. Now, both of these surfaces, which is lateral surface and medial surface, or external or internal surface, have got different nerve supply. We have to memorize. The inner side, that is which is lying towards the tympanic cavity, it is supplied through the branches of vagus nerve, supplied with the branch of glossopharyngeal nerve. So this inner side, which is a mucosal side of the tympanic membrane, the glossopharyngeal, which is supplying to it. So the mucous layer, which is a 9th cranial nerve, which is a glossopharyngeal, which has a branch, we call them a tympanic branch, which is supplying. Now coming to this particular region, which is cuticular region or the skin side. Now the skin side of the tympanic membrane is sourced from 2 different areas or 2 different cranial nerves. We have a nerve which is known as the vagus, which is the 10th cranial nerve, which is supplying to this external surface, along with the cranial nerve V, which is coming over here, the trigeminal, which is supplying to here. The trigeminal has a branch, we call them as auriculotemporal branch. So the auriculotemporal branch and the vagus, which is supplying to this particular external surface of the tympanic membrane, picking up the general sensation, touch, pain, temperature, and the vibration senses. On the inner side, it is picked up by this particular nerve, which is known as the glossopharyngeal nerve, which is the 9th cranial nerve. So this particular nerve, which is picking up or taking this particular sensation on these two regions, is a kind of important entity of the nerve supply of the tympanic membrane is concerned. You have to memorize. Now, coming from this nerve supply to the blood supply. Now, this particular tympanic membrane, once it soars, once it develops, it loses the most of this blood supply to it. It becomes really or relatively avascular. Now, that's why if you have a perforation, if you have an injury which ripped the tympanic membrane, it's hard to heal. So, a rupture of the tympanic membrane is hard to heal because it loses most of this blood supply to it. So, the middle ear cavity is supplied with some of the blood sources, which we will be coming again. But in an instance, the tympanic membrane itself is very less vascular. Only the margin little bit have got like a blood supply to it. More it moves to the central part, it is avascular. That's why I am saying once this thing is kind of like ripped off, broken, ruptured, it hardly heals. So you have to do surgery. You have to do patch-up surgery. That patch-up surgery, we call them as tympanoplasty. Now, coming to the clinical aspects. Now, if you have a tympanic membrane, which is having two different nerve supplies on the two different areas. Now, depending on the inflammations of the tympanic membrane, which is affecting to it from the inner side or external side, we have different nerve sensation or different nerves picking up this. So, you have an ear infection which is involving the external ear and that is moving towards this tympanic membrane. Then if you are feeling the pain sensation or any kind of discomfort, that is picked by the cranial nerve number 5 and 10. Compared to that, if you have some pathology inside the middle ear cavity, inside to it, then pain, temperature, sensation is picked up by that particular cranial nerve number 9, glossopharyngeal. That's the thing. So, we have two different nerves to pick up the sensation from different domains of this tympanic membrane. That's a clinical aspect, a clinical thing that you have to know or remember. Second thing, as I mentioned, a rupture of tympanic membrane is pretty common. Now, you tell me, we have two parts of this tympanic membrane. One is tense part, other is loose part. So, if you have a chance of trauma or anything, injury, hitting this particular side of your face or head region, and that force is transmitted on the tympanic membrane, which part of the tympanic membrane is more prone to get ruptured or perforated? The tense part or the loose part? The tense part or the loose part, if you are receiving any trauma, which part is more prone to have chances of perforation? Tense part, because it's tense. If you pinch there, it's going to just like have more impact than the loose part, which can accommodate some kind of stretch. So that's why this particular tympanic membrane, the rupture on them is more common on the tense part than that of the loose part. Now, if you have a rupture of the tympanic membrane, it's not going to heal on its own. So, it's going to make a hole itself if it is, or the hole remains there. So, you have to just like majority of the time, you have to patch it. And that particular patching may be putting some kind of membrane taking from elsewhere and placing it there. So, that kind of membrane placing on the tympanic membrane, we call them as tympanoplasty. Now, if you have a tympanic membrane which is ruptured, what is going to happen to the patient? What discomfort or the problem will be experienced by the patient? You have one tympanic membrane, say for example, right side is perforated. So, what will be the complaint of the patient? how do you expect the patient will be coming to your outpatient and you will be just like exploring that? What's the function of tympanic membrane, by the way? What's the function of it? Louder. What's the function of it? Tympanic membrane? It has no function or bizarre function. What is the function? I do not care. So what is the function of tympanic membrane? It's an 8th standard question. Did you pass the 8th standard or not? So what is the function of it? Vibration. What makes with the vibration? It catches up the sound wave and vibrates, and that vibration amplifies the sound. So if it is not vibrating well, if you have a hole in it, it cannot vibrate. So what the patient is going to experience? Deafness, thank God. So it may be just like a hearing less on the affected ear. Am I right? Am I right? No, right. The patient is complaining of not hearing well on the ear. কথা বললে শুনতে পাচ্ছি না। কি বলছে কি আবলতাবল বলছে যেমন এখন তোমরা শুনতে পাচ্ছ না। পারফোরেটেড টিম্প্যানিক মেমব্রেন কি প্রশ্ন শুনতে পাচ্ছি না তো উত্তর দিতে পাচ্ছি না তাই। So if you have a perforation of the tympanic membrane, the vibration will be diminished or absent. The patient is not going to hear the sound well, and there will be a partial or complete deafness depending on the affected side. If it is both sides, there will be bilateral deafness or partial hearing or complete hearing loss or something like that, right? So this is the thing which is going to be experienced by the patient. If you have a ruptured tympanic membrane, so functions, it moves with the response of the vibration, picking up the sounds, transmitting this particular thing from here to there is somebody else's job. Its function is to pick up the sound and vibrate. That's it. So this vibration will be dumped if you have the hole in it. So you have maybe hole involving in the central part, peripheral part, or maybe just like a complete loss, only the particular part which is known as tympanic sulcus on the bony socket where it is fitted, it may be there. It may be absent completely. So you have learned this development, right? Where from this tympanic membrane itself is developed from. Source, the entire source of the tympanic membrane. If it is arch or pouch or cleft or whatever, read books. And this is what we are talking about. The surface of this tympanic membrane externally is supplied with the 2 cranial nerves. This is the large supply which is coming from this cranial nerve number 3, which is the auriculotemporal branch of this V3. This blue one is supplied for the cranial nerve number 10. Now, so once you have done with the tympanic membrane, we can enter to the cavity of the tympanic region. We call them as tympanic cavity, also known as middle ear cavity. Now the middle ear cavity, as I mentioned, it has some contents to it. That means it has some structures living in there. And as well as it has some features which is present on the walls. So 2 entities on the middle ear we are going to talk. We are going to learn what are the contents there and related thing which is content is concerned to us. Second thing about the boundaries or walls. Now, how this particular cavity of this middle ear looks like. It has a hexagonal box. That means it has a 6 walls within it fitted there. So this middle ear cavity is fitted on the speechless part of the temporal bone and placed inside, and it has 6 walls. That means it has a floor, it has a roof, it has a middle wall, it has a lateral wall, it has an anterior wall and the posterior wall. And each of these particular walls are important, and you have to memorize what are the structures that are present in it. Alright, so this is the thing we are going to talk here now. First we talk about the contents of these region of the middle ear. The middle ear content here, the primary content is some bones which are present in it. So the bones which are present in it are very small. We call them as ossicles. You cannot call them as bones. We call them as ossicles. They are small minor bones. We call them as ear ossicles. So ear ossicles, how many of them are there? 3. Good. Good, bad and ugly. What are the names of them? Malleus, incus and stapes. Good. Now it looks like I am in a right class. Alright. So we have 3 bones present in it. Inside to ear, besides 3 bones, ear ossicles, we have 2 muscles present in it. So we have 2 muscles and 3 bones present in it, and along with some nerves and blood vessels which are present as a content of it. So bones you have mentioned. There are 2 muscles present in it. One muscle is related to one bone and other muscle is related to the another bone. So 2 muscles. One is known as tensor tympani, which is tensing the tympanic membrane. And other muscle is related to another bone which is making this bone strong fitted towards its attachment. We call them as stapedius. So these are the 2 muscles and the 3 bones which are present as a primary content. Along with we have the blood vessels, along with we have the nerves to supply to them. So if you have a nerve, if you have a muscle, you need a nerve to supply to them, right? So we have nerve to stapedius, we have nerve to tympanic membrane or tensor tympani to supply to it. Now coming back to your embryology class, which never happened actually. We have 2 muscles, we have 3 bones. So where from these ear ossicles are developed from and where from these muscles are developed from? Alright, hold a minute. I let go these particular bones. So muscles. So where from this tensor tympani developed from? Which pharyngeal arch or pouch? Is it arch or pouch? Arch or pouch? Pouch. Cleft. Cyst. Arch, okay. How many pharyngeal arches we have? 5, 4, 6. 1, 2, 3, 4, 6. 1 dropped out. 5 dropped out. Okay, quit the MBBS course. Lucky guy. Alright, first pharyngeal arch. It's the final answer. Tensor tympani is sourced from first pharyngeal arch. First pharyngeal membrane. Now you are changing the versions again. See? So you tell the patient you have Down syndrome, then you say no, you have diabetes. That's how you are going to teach. That's how you are going to treat. So you tell that it's coming from first pharyngeal arch. So first pharyngeal arch. I take it first pharyngeal arch. Okay, Ritik is sure. First pharyngeal arch. What's the nerve supply to first pharyngeal arch derivative? Cranial nerve number which? 5. Okay. So what's the other muscle we are talking about? Stapedius. Stapedius is sourced from which pharyngeal arch? Second. What is the nerve supply to it? Whichever the second derivative. 7. Right? So this is the thing which is you have to memorize. The muscle developed from which pharyngeal arch and supplied with these which cranial nerve and the actions of it. Besides this particular ear ossicles. By the way, ear ossicles you remember by any chance? Developed from which pharyngeal arch, which pharyngeal pouches? Okay, I remember. It will be taking day long standing over here. But coming back again, once again to this middle ear. It's a cavity, temporal bone, petrous part, which is lying between this particular middle ear, external ear and inner ear, which has a 2 cavity communicating with anteriorly with the tympanic, anteriorly connected with this nasopharynx by this particular tube, which call them as auditory tube. Backside the middle ear is having a connection as well with the mastoid bone. We call them as mastoid antrum. Through this particular opening, we call them as mastoid hiatus. Now this particular middle ear cavity we are talking here is a content over here, these 3 ear ossicle, malleus, incus and stapes. Along with we have 2 muscles which is placed inside to it. We call them as tensor tympani and stapedius. Now the stapedius and the tensor tympani source from 2 different pharyngeal arches, and they are supplied with the different cranial nerves. The stapedius source from the second branchial arch or pharyngeal arch, and nerve supply to the stapedius is coming to the cranial nerve number 7, whereas the tensor tympani, which is coming over here from the first pharyngeal arch, developed from the cranial nerve number 3, and number 5, which is the V3 divisions of this particular mandibular division supplying to it. Now, what are the attachments of this particular guy, which is known as tensor tympani? So this tensor tympani is attaching, one attachment is on the sphenoid bone and other is on the handle of the malleus. So we have the malleus, handle and neck of the malleus over here. We have this particular tensor tympani which is attached to it. So tensor tympani which attached to the handle of the malleus. Now what are the functions of the tensor tympani? If you split the name, you get the answer. It tenses. So it contracts, it pulls the tympanic membrane. When it pulls the tympanic membrane, it pulls the tympanic membrane towards the cavity side. It pulls the tympanic membrane towards the inner side. By doing it, it helps in dumping the vibration. If it is jarring too much, the tympanic membrane, it may rupture. So if you have exposure to loud sound, where you get the loud sound? In disco jockey, nightclubs or elsewhere, anywhere. If you are exposed to sudden high vibrating sounds, the tympanic membrane is going to and if it is doing that, it is going to rupture. To prevent that, we have this tensor tympani, it contracts reflexly and it pulls this inner side and it pulls and holds it so that it limits the vibration of the tympanic membrane. It's stabilizing that and dampens this vibration over there and can transmit the necessary voice which is related to it. So this is the tensor tympani, functions of it, contractions of it. Now coming to the other muscle which is also defense muscle, which is known as stapedius. So this particular stapedius inserted on this muscle, bone which is stapes, and it is the neck of the stapes. And once again its muscle contracts, it pulls posteriorly. That means it steadies the union of the footplate of the stapes on a membrane. We call them as the window, the oval window. It is doing a compression over there. So if you see this particular bone, we have the stapes which looks like this, stirrup. So this particular part we call them as footplate of the stapes. This particular footplate of the stapes is fitted on here. So if the stapedius contracts, it pulls this medially or it pulls it posteriorly on the back side. So this particular plate or foot plate of the stapes is steadied on the membrane on which this particular guy is sitting there, and it steadies that particular steadies this particular bone and again cut down the vibration sense which is coming on here. So whatever the sound that is hitting this eardrum picked up by this particular ear ossicle, transmitted through and put to the inner chambers of the ear, which is known as this cochlea or organ of Corti eventually. So this is the way this particular ear ossicles functions, malleus, incus and stapes, along with the muscles which are present to it and supplies to it. Okay, good. Now we are going to talk about the last part of this content, what am I going to see, going to talk here. Now walls of the tympanic membrane. So as I mentioned, the tympanic membrane cavity have got 6 walls in them: roof, floor, anterior wall, posterior wall, medial wall and lateral wall. The one wall we have already covered, the lateral wall. The lateral wall is covered by the tympanic membrane itself. So the tympanic membrane and the handle of malleus, which is covered already. So we are done with the one of the wall discussion on this part. So we are going to talk about the easy sections over here. Let's talk about the roof of the tympanic membrane. So the roof of the tympanic membrane is formed by the petrous part of the temporal bone. This piece of bone is there. We call them as tegmen tympani. So we have a piece of bone which is forming the roof, which is thin plate of the bone lying on the petrous part of the temporal bone, which is known as tegmen tympani, which is separating basically the brain with the cavity of the middle ear. So between the brain, meninges and the blood vessels, this particular piece of the bone here, we call them as tegmen tympani, and this particular thing is separating from this middle ear cavity with this. So this is the functions of the roof. Now, we have a clinical importance to this roof. In case of the kids, this tegmen tympani piece of the bone may be membranous. It haven't ossified yet. Now, what happens if it is membranous? What's the big deal about it? The big deal is if the kid have got somehow contracted a middle ear infection, that infections from the middle ear can be transmitted through the membranous or porous tegment tympani, which is yet to ossify. So that's why if you have a kid newborn or just like 1 year old kid having kind of ear infection which has affected this particular middle ear, there are chances of spread of infection from ear to brain. So this is the importance of knowing the roof. In adults, it's kind of plate, but again, it's a very thin plate. It breaks. If it breaks, it has a communications with the cavity of the middle ear with the brain and can transmit the infection in the same way. That's the way it's happening. Especially, it happens in case of the trauma, head trauma. Most commonly happens during the punch games, the boxers game, which has a particular punch which is known as the uppercut. So those things which makes this particular thrust go on the tegment tympani, breaks it, and if the tegment tympani breaks, we have a very grave complications may be coming out if it not corrected. Okay, so this is the thing you have to know about. Other thing, this particular roof is pierced by the petrosal nerve. There is a nerve which is piercing the petrosal nerve. We'll be coming again about the petrosal nerve. Coming from the roof to the floor. The floor of this tympanic membrane is formed by the jugular fossa. We've seen this particular jugular fossa in this particular bone which is known as occipital bone. We have this jugular foramen. Next to the jugular foramen, we have the jugular fossa. That particular jugular fossa lodges the bulb of the internal jugular vein. So that particular floor of this particular region on this particular tympanic cavity, also known as middle ear cavity, is formed by the bony jugular fossa. And it separates the tympanic cavity with this bulb of the internal jugular vein. Now this particular region, floor, is pierced by a nerve which is known as tympanic branch of the 9th cranial nerve. And this is what we have to memorize. You have to remember this piece of information about this. So whatever I am writing here, if you write those few words, how many words I am writing? Two sentences, barely. If you write these words only, that is enough for you to get a good score. Besides the giving a schematic picture with addressing it with the proper clinical things. So this piece of information we want from you as an answer script. We are not asking you to talk about a history or geography about it. Just piece of information which is relevant, which is important to us. Now, coming from the roof and the floor, these are the easiest part. Then moving to this wall which has an important feature, the medial wall. Now this particular medial wall itself could be given as a part of the question. So medial wall elaboration is a kind of important thing that you have to understand, that you have to prepare. The medial wall or narrate the features of the medial wall of the tympanic cavity itself could be a short or itself could be a part of the question rather than discussing every wall. Now, first thing first. First thing, on the medial wall we have a bulge. So what is the medial wall? Which is lying opposite to the tympanic membrane. So medial wall at the center we have a bulge and a bony bulge. That bony bulge we call them as promontory. And that particular bony bulge is created by the cochlea which is present in the inner ear. But anyway, we have a bony bulge, we have to know the name of that, promontory. And this particular promontory is covered or carpeted by the mucous membrane which is the middle ear has. Now this particular, on this promontory we have a ramification of a nerve plexus. That nerve plexus which is ramifying or growing there, making a nest, we call them as tympanic plexus. Now how this tympanic plexus is formed, we are coming again. So we have a bulge, promontory. On a bulge we have a plexus of nerve network, we call them as tympanic plexus. Now on the top of this promontory and below this promontory we have two holes there. On the top of the promontory we have a hole which is oval, and below we have a hole which is the round hole. So the top of this promontory we have a hole, we call them as oval window. That's oval window which is known as fenestra vestibuli. That particular oval window is covered or patched by the foot plate of the stapes on which it is staping, and that particular thing which is sitting on it covering this particular window, the oval window. Under to the promontory we have another window, we call them as round window, which is a smaller one, we call them as fenestra, which is a fine small hole, cochleae. So which is covered by a membrane, we call them a secondary tympanic membrane. These are the two features that you have to remember. A bulge promontory on which there is a ramifications of a nerve, tympanic plexus. On the above and below we have two holes, oval hole and round hole. Oval hole which is covered by a foot plate of the stapes, and the round hole is covered by a membrane, secondary tympanic membrane. That's it. And to it we have a facial nerve crossing over. We have the facial nerve coming down from the brain, getting inside the stylomastoid foramen to reach to this particular parotid gland. So when it is coming down through the brain, it runs an oblique course. So on the top of this particular oval window, we have an oblique course or oblique canal, which is traveling through it, is having facial canal through it, having a facial nerve. So this piece of information that you have to collect here about this medial wall. As I mentioned, this particular feature itself is a short nerve. You have to write down this piece of information this much. That's it. With a picture, schematic picture, which I am going to show very soon. If you know the theory, then you can talk about. So this is about the medial wall. And what is the medial wall? Opposite to the tympanic membrane. That means if you throw the light, that's a lateral wall of the tympanic cavity. That means the lateral wall of this particular tympanic membrane you are seeing. You hear the tympanic membrane, then if the light directly goes, that will be falling on the medial wall. And on the medial wall, you are going to see these features. That's it. Now, other features, anterior wall. This particular anterior wall, which is present on the anterior side. So, anterior side, it has one thing which is present. We have an artery going to the brain, which is internal carotid artery. That is running through a canal, the canal for internal carotid artery. If you go back to the base of the skull, many a times we talked about that. Many a times we probe through it and ask about what is this and what runs through that. That's a cavity or the canal for the internal carotid artery, which goes through the brain, supplied to the brain. So that particular canal, which is having a vertical part. So the vertical part of this internal carotid artery is present on the anterior part. And besides that, we have two canals or two tunnels lying on top of them. There are two tunnels that is transmitting two structures. We have two tunnels, one above, one below. So the above tunnel is transmitting the tensor tympani muscle, which is coming from the sphenoid, the wing of the sphenoid, and getting inside to the angle of malleus. And that particular muscle is transmitting on the above canal and below canal, which is lying to it, transmitting a tube which is known as the auditory tube, the Eustachian tube. So, anterior canal, anterior wall, which has got two features. One is the carotid canal, which is lying on the bottom side. Above side, we have two tunnels. One is transmitting a muscle, one is transmitting a tube through it. That's it. Coming to this part, which is known as the last part, which is concerned, the posterior, which is lying on the back side. That means the posterior side is the mastoid. So we have the mastoid bone. Now this above to this region, above this particular space, posterior wall has an opening to communicate with the mastoid, which is a part of this temporal bone. So it is connecting with this upper part, which is hiatus. We call them as hiatus mastoid or mastoid hiatus, which is communicating with the cavity of the mastoid. And on the back side, we have this facial nerve, which is going down, having an oblique course and dropping down between this particular mastoid process and the styloid process, which has a hole there, stylomastoid foramen. So that particular facial nerve has a vertical course and an oblique course. Oblique course lying on this medial wall, then it is dropping down vertically and running out of this thing, which is lying on the posterior wall and the mastoid wall. So these are the features that is present in it. So that is easy. Now this is the thing that you are addressing. This picture you have to memorize. This picture you have to follow all the time. If you just like following it, pretty easy. You find the roof, that's a tegmen tympani. It's separating from the brain, and that is pierced by the petrosal nerve. You find the floor that has that vein, which is bulb of the jugular vein, that is pierced by a nerve, tympanic branch of the glossopharyngeal nerve, which is piercing through it. The tympanic membrane, you cannot see it here because that is taken out, and you are seeing the opposite wall of the medial wall. The medial wall opposite, which is opposite to this particular lateral wall, which is the medial wall. So this is the medial wall which you are seeing here. This is the medial wall. What is the feature that is present in a medial wall? A bulge. This is the bulge we are talking here. This bulge, we call them as promontory. On top of promontory, one hole, oval hole. On here sits the footplate of the stapes. Below hole, round hole. In here, secondary tympanic membrane. In the promontory, we have the plexus of the nerve. We call them as tympanic plexus. Now, in here, we have an oblique course of the facial nerve, which is running on top of this oval window, which is the facial canal, which is having oblique course coming down the frame, then dropping down to the posterior wall. This is the posterior wall. This is the anterior wall, roof and the floor. So this particular region, oblique region, we have this particular region, oblique part of the facial nerve running over here on this top part. So this is a feature we just talked about. So if you go back to this particular medial wall, we just mentioned about these few things about it. Promontory, oval window, round window, prominence of the facial nerve. That's it. With the schematic pictures like this. Coming to the anterior wall, which you are seeing here in this region. Anterior wall is this wall. Lower part, we have the facial nerve. Lower part, we have this particular carotid artery, which is running through the carotid canal. The vertical part, then it's turning down. So this part, we have this internal carotid artery. Upper part, we have two tunnels to transmit two structures. Upper tunnel, lower tunnel. Upper tunnel transmitted tensor tympani muscle. Lower tunnel, auditory tube. This is the anterior wall, features which is present in it. Posterior wall, which you are talking here. The upper part has a hole. We call them as hiatus, which is known as mastoid hiatus. Communicating with back side is the mastoid bone. This is the mastoid bone. So cavity of the mastoid bone is communicated through this hiatus. So mastoid hiatus with the communication on the upper side over here. Lower side, bottom side, we have this nerve which is coming over here. Same facial nerve, which is a vertical sign. What the facial nerve is doing? It is getting down out of the brain. Where from it is coming? Between these bony projections. Styloid process, mastoid process. Between here, we have the foramen here. We call them a stylomastoid foramen. It is getting out of the brain, getting inside the parotid substance. This is how it is going down. That's it. These are the features. Now, posterior wall have got a prominence over here. We call this prominence as pyramidal eminence. This pyramidal eminence is an attachment site of this small muscle. This small muscle is stapes. So this stapedius is attached to the posterior wall, pyramidal eminence, and anchored to the foot plate, or anchored to the neck of the stapes. And it is steadying the grip on the oval window which is present. And this particular stapes is supplied by the nerve, which is nerve to stapes, which is a branch of facial nerve, which is coming from somewhere here and supplying through it, piercing here, a branch coming here on the posterior wall, which is the chorda tympani. As I mentioned, the chorda tympani, which is a facial nerve branch going to supply the tongue, taste sensation, which is a spatial sensation, crossing across the tympanic membrane. If the tympanic membrane was here, it is crossing across the tympanic membrane. So whenever I talked about tympanic membrane, it is layered between these two layers. Tympanic membrane, you cannot see it here because it's present on the lateral wall. It's ripped, that's why you are seeing this. Good so far? So this picture and these one or two liner things basically narrates everything and emphasize on the medial wall, the features of them. Now, the last thing before we leave on this medial wall cavity, this region which has a plexus of the nerve. We call this plexus of the nerve tympanic plexus. Who makes this tympanic plexus? You see a nerve which is coming here, which is a tympanic branch of the glossopharyngeal nerve. What is the function of glossopharyngeal nerve inside the inner ear? It supplies the sensation in the inner ear. Inside the middle ear. So it supplies the mucosa with the sensation. What sensation? Touch, pain, temperature sensation. Picking up sensation from the middle ear. As well as it's supplying the inner aspect of the tympanic membrane. So sensory supply to this particular region. So this tympanic branch of the glossopharyngeal nerve comes over here, ramify over here, makes this plexus, which is known as tympanic plexus. Along with it, joins a nerve coming from this nerve, which you are seeing here. This is what? Internal carotid artery. Surrounding the internal carotid, it has a nerve. We call them as sympathetic chain. That sympathetic chain shoots a nerve here, which is known as caroticotympanic branch, which also comes over here, joins with this glossopharyngeal nerves, ramifies over here, and from here shoots a nerve which is known as lateral petrosal nerve. That lateral petrosal nerve goes to supply to the secretomotor supply to this parotid gland. So this particular region, we have to just like have an idea about what this particular plexus is made of. The first thing you have to memorize this is made of glossopharyngeal nerve. If you cannot remember the sympathetic chain or sympathetic fiber, it is well, nobody is bothering about. Please do remember about this particular plexus which is present in here, which is the tympanic plexus. Who makes this plexus? Cranial nerve number 9. What the plexus is doing there? It's giving the sensory supply to the middle ear cavity, the mucosa over there. That means touch, pain, temperature sensation, infection there, pain there, picked up by this. That's it. Okay? So this is about these different walls and very little features involving them. About this middle ear, about this walls, contents and everything. That involves the clinical thing. The lateral also I have mentioned. So prepare this as an exam short note with these few things about this middle ear cavity. Now this middle ear cavity receives the blood supply from two sources, coming from the maxillary artery branches principally, and it is coming from the occipital artery, which is going to supply the cavity of the middle ear, the muscles and the soft tissue structure present in. So this is about the middle ear cavity. We are not going to talk about this auditory tube because I do not have the time to talk about it. So auditory tube may be later in a separate session. So that's it for the day. Have doubts, queries? No? Alright, fine.
What is this? This is the cricoid cartilage. So what is the typical feature of cricoid cartilage is that it is a complete seed. It is a signet ring appearance. Okay. Trachea the baki cartilage gulo ki incomplete or C-shape except this one, that is the cricoid cartilage. What about this one? It is the epiglottis. It is also one of the cartilage of the larynx. Okay. And in between that you could get this membrane that is known as thyroid membrane. So from the hyoid bone to the thyroid cartilage. Okay. And what is this muscle? That between the thyroid cartilage and the cricoid cartilage. So this muscle is cricothyroid. Cricothyroid. Okay. So for knowing about the larynx, you have to know it is featured about 9. Okay, how 9? It has got 9 cartilage. Okay, so among them 3 are unpaired and 3 are paired. So name the unpaired. Unpaired কি আছে? Thyroid, cricoid, আরেকটা হচ্ছে epiglottis. তিনটে দেখা যাচ্ছে, রাইট? তাহলে paired কোনগুলো আছে? Peritinoid, corniculate and cuneiform. ঠিক আছে? So total there is 9 cartilage. Then আর কি 9 আছে? What is the length of larynx? Length I told it is about 9. Okay, so in case of male it is 9 5s of 45 millimeter, and in case of female it is 9 4s of 36. Okay, easy to remember. ওর ক্ষেত্রে হচ্ছে 45 millimeter male আর ফিমেলের ক্ষেত্রে হচ্ছে 36. Okay, okay, আর কি আছে larynx-এ? Why larynx is important? What is the function of larynx?
Voice.
Contain the voice box. RT, it is a part of the respiratory tract. So could you elaborate the respiratory tract? কি আছে আমাদের? প্রথমে nasal cavity, then nasopharynx, then laryngopharynx, then no oropharynx won't come. Oropharynx will be part of digestive tract. ঠিক আছে? laryngopharynx ছিল, তারপরে ট্রেকিয়া, বুঝতে পেরেছিস? ট্রেকিয়া, তারপর দুদিকে দুটো ব্রঙ্কাস, দেন লাংস, প্রাইমারি, সেকেন্ডারি, টারশিয়ারি, ব্রঙ্কিওল এন্ড ফাইনালি অ্যালভিওলাই। ঠিক আছে? Okay, so see, larynx is a space that communicates above with the laryngeal part of the pharynx, যেটাকে আমরা বলছি laryngopharynx. Okay, and below it is continuous as a trachea. So this is the laryngopharynx. Okay, this part. So what is happening while you are taking any food? যখন খাবার খাই কি হয় তখন pharynx আর larynx দুটো কি করে rise up, it used to rise up when the food enters here. So at that time this epiglottis is closed, closing where the glottis is, opening of the larynx, এটাকে ক্লোজ করবে so that any food could not enter the larynx. ঠিক আছে, তার মানে দুটো সুইং ডোর আছে, একটা হচ্ছে এটাকে কি বলছি সফট প্যালেট। যখনই কোনো খাবার মাউথ দিয়ে যাচ্ছে তখন সফট প্যালেটের এই ডোরটা কোথায় যাচ্ছে, উপরে চলে যাচ্ছে। ঠিক আছে, তাহলে এখানে খাবারটা যেতে পারছে না ন্যাসোফ্যারিংসে। ঠিক আছে, আর এটাতে কি হচ্ছে, যখন এপিগ্লটিসটা ক্লোজ করে তখন কিন্তু খাবারটা একইভাবে ল্যারিংসে ঢুকতে পারে। ঠিক আছে, তো অ্যাপার্ট ফ্রম দ্য রেসপিরেটরি প্যাসেজ অর্গান ইজ কনসার্ন উইথ দ্য ভয়েস বক্স। ভয়েস বক্স কোথায় আছে, এই দেখ, এটা হচ্ছে ভোকাল কর্ড, রাইট? সো ভোকাল কর্ড হাউ ইট ইজ ফর্ম, এটাকে বলছে গ্লটিস। গ্লটিস ও ইট কল গ্লটিস। ওকে, সো নেক্সট ওয়ান ইউ কুড সি দ্যাট ইজ দ্য what are the relation, what are the anatomical relation. It is covered anteriorly by infrahyoid muscle, anteriorly by the infrahyoid muscle. Could you name some anterior triangle chilo infrahyoid muscle ki ki ache bol, thyrohyoid, sternohyoid, sternothyroid, omohyoid, হচ্ছে infrahyoid muscle, right? আর কি মাসেল পাবো, the muscle of the pharynx, that is inferior constrictor. আর কি মাসেল আছে pharynx-এর, superior constrictor, middle constrictor, এগুলো থাকবে না। আর তিনটে মাসেল, salpingopharynx, very good, salpingopharyngeus, palatopharyngeus, stylopharyngeus। ঠিক আছে, তার মানে টোটাল হচ্ছে যত pharynx-এর মাসেল they will also support, okay, this larynx, and posterior to the larynx we know there is esophagus. Okay, so here is one maneuver, this thing you should know in case of emergency condition. Okay, so emergency condition e ki korche, tokhon amra intubate korchi, right? So kothay eta ki instrument er naam ki laryngoscope. Okay, laryngoscope diye mainly jara ICCU te thake tara to jane, ঠিক আছে, তারা যদি না জানে তাহলে আমাকে অ্যানাস্থেসিস লাগবে। ঠিক আছে, বিকজ দে আর ভেরি ইউজ টু উইথ দিস প্রসিডিউর। তো ওরা কি করে, মেনলি দে উইল গো আপ টু দ্য ল্যারিংস। ওকে, মেন থিং ইজ দ্যাট দে সেপারেট দ্য ল্যারিংস অ্যান্ড দে পুট দ্য চ্যানেল ওভার হেয়ার আপ টু দ্য ট্রেকিয়া। ল্যারিংসটা না পেলে ট্রেকিয়া পাবো না, তার মানে আমাকে এমার্জেন্সি ইন্টিউবেশন কখন করতে হবে, when there is tremendous breathing problem, পেশেন্ট আর নিঃশ্বাস নিতে পারছে না, তো অ্যাট দ্যাট টাইম করবো। আর কখন করবো, কোনো ওটি হচ্ছে, ওটির সময় করতে হবে। কেন, আনকনসিয়াস পেশেন্ট, সে কোনো কিছু খাবার রিগার্জিটেড বা অ্যাসফিক্সিয়া হতে পারে। ঠিক আছে, তো এই দুটো কেসে তখন আমি ইন্টিউবেশন করবো। অ্যান্ড দিস প্রসিডিওর রিমেম্বার ইট ইজ নোন অ্যাজ সেলিক্স ম্যানিউভার, ইট ইজ নোন অ্যাজ সেলিক্স ম্যানিউভার। কোথায় দিচ্ছি, উই কুড পুট প্রেশার অ্যাপ্লাইড টু দ্য ক্রিকয়েড কার্টিলেজ, ক্রিকয়েড কার্টিলেজের উপর প্রেশার অ্যাপ্লাই করা হয় এবং টু অক্লুড দ্য esophagus, main intention to occlude the esophagus. Okay, so it's a emergency intubation. Okay, so what are the function of larynx, ki function ache, number 1 is phonation, second is respiration, these two you know, right? Another one is protection, it is acting as a sphincter or valve for the lower respiratory tract so that no foreign body could enter to the lungs. ওকে, প্রথমে কি হয়, যদি ফরেন বডি চলে যায় জ্বরটা কি হবে, কাফ। ওকে, দেয়ার উইল বি ট্রিমেন্ডাস কাফ রিফ্লেক্স। ওকে, অ্যান্ড ডিগ্লুটিশন, সো যখন এনি ফুড আপওয়ার্ড ডাউনওয়ার্ড মুভমেন্ট হয় ডিউরিং দ্যাট larynx also help. So these are the four functions: phonation, respiration, protection and deglutition. Okay, so what is the extension? Extension is from C3 to C6. So this is the extension C. This is the C3, that means root of the tongue up to the trachea, root of the tongue up to the trachea, right? So this is the extension of the larynx. এটা কিন্তু এমসিকিউ আসে পাবে, প্রচুর বার এমসিকিউ পাবে। এক্সটেনশন অফ larynx মনে রাখবি C3 to C6. Okay, next one is Adam's apple. It is nothing but the prominent thyroid cartilage. তো এটা কেন মেলের ক্ষেত্রে কেন প্রমিনেন্ট হয়, 120 degree angle. Okay, angle is the one factor, anything else? So at first তুই যদি একটা ছোট বাচ্চা বা একটা উইমেন বা একটা মেল যাকেই দেখিস বিফোর পিউবার্টি দ্য ভয়েস ইজ সেম, আফটার পিউবার্টি কি হয়? দেয়ার ইজ এ স্পিচটা চেঞ্জ হয় কাদের? মেলে, because of this Adam's apple. Okay, because it become more enlarged so that the voice could be produced. Okay, so what would be the typical voice of male? It would be loud, low pitch, and in case of female it is high pitched voice, right? For babies also it is high pitched, right? The cry, everything is high pitched. Okay, so this is the length of larynx in case of male. Okay, so 9 5 is 45, and in case of female it is 36. Okay, okay, so this is the Adam's apple or the thyroid cartilage. Okay, so see here, what are the things we could see? First one, what is this? This is the epiglottis, right? What is this membrane? From thyroid cartilage to hyoid bone, thyroid membrane. So see which structure is piercing here. This is the MCQ. Internal laryngeal branch of recurrent laryngeal nerve. Okay, and there is another branch of vagus, that is the external laryngeal nerve. So both this internal and external branch, it is giving all the muscle of the larynx. Okay, and if you see the right recurrent laryngeal nerve, you could see it is more in length. Why? Thorax courageous cannot right recurrent laryngeal nerve of vagus is more longer. Can you answer? Because it hook around which structure? Right subclavian artery. Okay, right subclavian artery. But the left one is running parallel. Okay, we will see the next picture. Okay, so these are the cartilage of larynx. Okay, unpaired কি আছে? Cricoid, epiglottis and the thyroid. Cricoid, epiglottis, thyroid. Paired কি আছে? Arytenoid, corniculate and cumulate. So could you tell me which one is the hyaline, which one is the elastic? There are two type of cartilage, hyaline and elastic. Hyaline cartilage could be converted to the bone. So which are the hyaline? Cricoid the hyaline.
thyroid the hyaline.
Hyaline to correct. Remaining all are the elastic. But remember this arytenoid. Apex of the arytenoid is hyaline. Okay. So 3 are the hyaline and remaining 3 are the elastic. How you remember? E and C is looking same. Small letter E and C are same. So epiglottis, corniculate and cuticle, these are the elastic. Okay. So see here, what is this? This is the epiglottis. So if you see the larynx from outer surface, first thing you will get, this is the thyroid cartilage and this is the cricoid cartilage. Okay. And see the inside, this is the epiglottis. What is this space? This place টাকে কি বলে? Common site for lodgement of fish bone. মাছের কাটা খেলে ওখানেই আটকে যায়। Pyriform fossa. It is pyriform fossa. So you could see this is the pyriform fossa. What is this? This is the arytenoid cartilage. And you could see apex of the arytenoid cartilage ke ache? Corniculate cartilage. Okay, and this is the cricoid cartilage. So this is the external picture, this is the internal picture. Now here what is interesting, see how the fold is created one kind of gap. This is nothing but the rhima glottis. Rhima glottis. How it is produced? Superiorly there is two fold, that is known as vestibular fold, and inferiorly there is vocal fold. So these two are producing one space. So what is the importance of this space? During breathing or phonation, very inspired air are going through this. Okay, at that time it is open, and whenever it is closed during expiration, or when the already voice is produced. Okay. Rhima glottis. Rhima glottis. And this part is known as glottis. Above is supraglottis, below is subglottis. These are the part of the larynx. Okay. Now what are the cartilage? This thing, thyroid, cricoid and arytenoid. Apex of the arytenoid. Base of the arytenoid is elastic. Okay. These are the hyaline cartilage. They tend to ossify above 25. যেমন তোদের কারো এখন ossify হয়নি, তাই তো? So above 25, the corniculate, cuneiform and epiglottis are the elastic cartilage. Okay. Now about the thyroid cartilage, it is the largest cartilage. Okay. And it has got two laminae. Both sides যেটা দেখা যায় সেটা কি বলছি laminae. And in between there is a V-shaped projection that is known as Adam's apple. The tubercle. Okay. And if you notice the thyroid cartilage, above there will be one notch that is known as superior thyroid. Below another notch that is inferior thyroid notch. Okay. And with the hyoid bone I have shown you, there is thyrohyoid membrane. Okay. And additionally there are two ligaments. One median thyrohyoid ligament and two lateral thyrohyoid ligament. Okay. And there is one joint, only one joint you have to remember, that is cricothyroid joint. So cricopharyngeus and thyropharyngeus is a part of which muscle? Pharynx বলছে সোরা? Is a part of inferior constrict. Okay. Cricopharyngeus and thyropharyngeus. Why I mentioned? There is formation of one diverticulum that is known as Jenka diverticulum. What will happen if somebody is taking the food used to get lodged over there. Okay, if somebody has that. Okay, so just study the Jenka diverticulum. Okay. Okay, now cricoid cartilage, it is signet ring shape. Only one cartilage in the trachea which is complete. Okay, only single cartilage in the trachea which is complete and located directly below the thyroid cartilage. Okay, and it is connected to the trachea inferiorly and it is the only complete ring of the trachea. Okay, cricoid cartilage. Now this may comes as short note. What is rhina glottis? Okay, so it is the narrowest anteroposterior cleft of the laryngeal cavity. Now it left the transverse cleft, not left the anteroposterior cleft. Okay, and length koto? In case of male it is 24 millimeter and in case of female it is 16 millimeter. Okay, and divided into two parts. It has got intramembranous and intracartilaginous. Some part are membranous, anterior 3/5 and posterior 1/5 is cartilaginous. So membranous part is more. Okay. So see, this is the rhina glottis. Okay. So you could see there are two folds. One is vestibular fold, another one is vocal fold. So this place is known as rhina glottis. Okay. So what are the boundary? Anteriorly ki ache? Angle of the thyroid cartilage. Anteriorly jodi dekhi angle of the thyroid cartilage. Posteriorly ki thakbe? Intrathyroid fold of mucous membrane. And laterally vocal fold, that is anterior 2/5 and arytenoid cartilage on the posterior 1/5. Okay. So what is happening with the shape of the vocal cord in along with the movement? কি চেঞ্জ হচ্ছে দেখ, প্রথমে বলছে কোয়াইট ব্রিদিং, ডিউরিং স্লিপিং অর কোয়াইট ব্রিদিং কি হচ্ছে ইন্টারমেমব্রেনাস পার্ট যেটা আছে, এইমাত্র হলো মেমব্রেনাস পার্টটা বেশি বড়, so that would be looking like a triangle. You could see it is a triangle, and intercartilaginous part it is looking like a quadrangle. ঠিক আছে, then full inspiration you could see this is a looking like a diamond. Okay, diamond shape due to abduction of the vocal cord. High pitched voice কি হবে? linear chink adduction of the vocal cord. So during phonation খুব জোরে যদি কেউ আওয়াজ করে then it will look like a linear chink. Okay, and whispering কি হবে? inverted funnel shape. See, this part is large, this part is small. Okay, so this thing you have to explain if this comes. Okay, movement of the vocal cord along with the different movement. Okay, so here we could see during quiet respiration, see how the shape during forced inspiration, so it is like this. So তোদের কি কোশ্চেন থাকতে পারে ইমেজ বেসড কোশ্চেন, ঠিক আছে? সেক্ষেত্রে কিন্তু এই ধরনের পিকচার থাকতে পারে, ঠিক আছে? একটা এমসিকিউ দিয়ে দিল এটা কি ধরনের বল, জাস্ট ইউ হ্যাভ টু রিমেম্বার দ্য ডিফারেন্ট শেপ। ওকে, okay, so extrinsic muscle already I have mentioned. These are the muscle of the pharynx: stylopharyngeus, palatopharyngeus, salpingopharyngeus, thyroid and sternopharyngeus. So could you tell me the nerve supply of pharynx? এই উইকে তোদের হেড নেক শেষ, হেড নেক শেষ মানে ব্রেনটা শেষ হয়নি, ব্রেনটা হবে। প্যারেন্টসে কি নার্ভ সাপ্লাই বুঝো? So why it is important? The nerve supply, arterial supply, venous drainage, all are coming from thyroid. Okay, as because thyroid is situated just proximally. Okay. So intrinsic muscle kiki ache? You have to know what is the function. Okay, thyroepiglotticus. It opens the laryngeal inlet. Okay, and oblique arytenoid and aryepiglotticus closes the inlet. Just the reverse. Thyroepiglotticus and oblique arytenoid and aryepiglotticus. Very difficult this one. So see, this is the oblique arytenoid, how it is placed, and this is the transverse arytenoid and posterior trichoarytenoid. So this is showing the same thing. So see, this is the oblique arytenoid. For the vocal cord, trichothyroid and vocalis tenses the vocal cord. এগুলো কিন্তু এমসিকিউ আছে, ঠিক আছে? So trichothyroid and vocalis tenses the vocal cord, whereas thyroarytenoid relaxes the vocal cord. So that's why the trichothyroid এইমাত্র আমি কি বললাম, it is supplied by external laryngeal nerve. So ওটা যদি প্যারালিসিস হয় কি হবে? Vocal cord is remain open. Okay, tense করে, ওটা টেন্স করতে পারছে না, মানে রিল্যাক্সড অবস্থাতেই থেকে যাবে। So just counteracting muscle is thyroarytenoid. So see here they have shown other muscle like transverse arytenoid and thyroarytenoid. Arterial supply kiki ache? Superior laryngeal artery is a branch of superior thyroid. Again it's a branch of external carotid artery. Okay, and nerve, the nerve is followed, that is superior laryngeal nerve. Again it is a branch of vagus. Okay, inferior laryngeal artery is a branch of inferior thyroid. So you have to just remember the blood supply of thyroid. Okay, and thyrocervical tract. যদি কারো থাইরয়েডের টিউমার হয় বা ক্যান্সার হয়, রাইট, থাইরয়েডটা কেটে বার করতে হবে, সেক্ষেত্রে কোন নার্ভ অ্যান্ড আর্টারিটাকে আমরা প্রিজার্ভ করব? থাইরয়েডের উপরে একটা নার্ভ অ্যান্ড আর্টারি আছে, নিচে একটা নার্ভ অ্যান্ড আর্টারি আছে, রিলেশন কুটু নেমলে কি কি আছে? So this is the assignment for you. Okay, which structure could be found at the superior and inferior border of the thyroid? One nerve, one artery, again one nerve and one artery, and you have to preserve that. Basically the nerve. Nerve যদি কিছু হয় কি হবে? অ্যাপোনিয়া। কেন? যদি থাইরয়েড বেশি হলে সেখানে মানে হোর্সনেস অফ ভয়েস হয়, বুঝতে পারছিস? এখন লিঙ্কটা সেম নার্ভ সাপ্লাই, তো সেই জন্য হোর্সনেস অফ ভয়েস। Which structure is present superior and inferior border of the thyroid gland? Or which nerve and vessels are related to superior and inferior border of thyroid gland? Okay. So inferior laryngeal artery is a branch of thyrocervical trunk. Okay, and it follows the recurrent laryngeal nerve. তাহলে inferior laryngeal artery এর সাথে কি আছে? recurrent laryngeal, আর superior laryngeal artery এর সাথে কি আছে? superior laryngeal nerve. So see here, this is the superior thyroid. Okay, and from the thyrocervical trunk, this is the inferior laryngeal. Okay, then next is the venous drainage. So superior laryngeal vein, again from the superior thyroid vein, and inferior laryngeal vein from the inferior thyroid. Okay. what are the lymph nodes? First of all, there is upper deep cervical, lower deep cervical, and pre-tracheal, trachea सामने আছে, pre-tracheal and pre-laryngeal. Okay, these are the four group of lymph nodes. যদি কারোর ক্যান্সার মানে বলে যে তোমার ক্যান্সার হবে, তখন যদি কেউ বলে যে অপশন নিতে কোন ক্যান্সার নেবে, তখন বলতে হবে গ্লটিক ক্যান্সার. So the mucous membrane of the larynx above the vocal cord is supplied by internal laryngeal nerve, and below the vocal cord is external laryngeal nerve. So this is the demarcation. Vocal cord is the demarcation. Above is internal laryngeal, below is external laryngeal. Okay. So see here, this is the nerve, right? What is this? This is the recurrent laryngeal. ঠিক আছে, রেকারেন্ট ল্যারিঞ্জিয়াল উপরে যেটা দিচ্ছে সেটা হচ্ছে ইন্টারনাল ল্যারিঞ্জিয়াল, নিচে যেটা দিচ্ছে সেটা হচ্ছে এক্সটার্নাল ল্যারিঞ্জিয়াল. কেন নামটা হলো এরকম ইন্টারনাল এন্ড এক্সটার্নাল? দেখ, এটা ইন্টারনাল বর্ডারে যাচ্ছে আর এটা এক্সটার্নাল বর্ডার অফ ল্যারিংসে যাচ্ছে. সো দ্যাটস ওয়াই ইন্টারনাল অ্যাবাভ এক্সটার্নাল বিলো. ওকে, সো দিস ইজ দ্য মোস্ট কমনেস্ট এমসিকিউ you will get about 100 times. Okay, all of the muscle of larynx is supplied by recurrent laryngeal nerve except cricothyroid, which is supplied by external laryngeal nerve. How you remember? Small letter C and E look similar. So cricothyroid is supplied by external laryngeal nerve. Okay. Numerous times it is repeated. Okay. So now the clinical anatomy. What are the things? First thing is foreign body entrapment. Jodi hoy, what will happen? There will be vigorous cough, and ultimately if it is not cleared, so there will be suffocation and even death. So you all know choking. And laryngitis is what? Inflammation of larynx. So what will happen? There will be accumulation of fluid, and it is causes hoarseness of voice. So this is the first aid you have to do as a doctor. Okay, so 5 times you have to thrush. Okay, then just see, he has already closed the finger. Then 5 times push from front. Okay, vocal cord is responsible for the production of voice. Okay, so if the recurrent laryngeal nerve is paralyzed, so what will happen? There will be hoarseness of voice. So why it is paralyzed? I told it has got long course because it hook around the right subclavian artery. But whereas left is straight or parallel. Okay, left recurrent laryngeal nerve. Okay, so here nothing is written. So first thing you have to see about unilateral. That means one-sided recurrent laryngeal nerve pulse. What will happen? Hoarseness of voice. Bilateral হলে কি হবে? অ্যাপোনিয়া, কথা বলতে পারবে না, রাইট? And finally যদি কমপ্লিট প্যারালাইসিস হয় তখন death. Okay, so that much serious condition. Okay, breathing is impaired and phonation also would not occur. So কিরম থাকবে? দেখ, one thing you have to see how would be the movement of the vocal cord. In bilateral paralysis, it is between adduction and abduction, in between. Okay. So here they have shown, see, with paralysis it is completely closed. Unilateral paralysis, half side is open, and bilateral paralysis is completely closed. Okay. So next one is when it is partially paralyzed or damaged. So that is fully adducted position. Okay. And it occurs bilaterally in the rhima glottis, so it is completely closed. And if it is completely closed, it is emergency condition. Okay, needs surgical intervention. The same thing. Next one is what is singer's nodule. যারা বেশি গান করে, যারা প্রচুর কথা বলে, তাদের কি হয়? ভোকাল কর্ডে ছোট টিউমার টাইপের থাকে. It's a benign tumor. Okay, so that is a singer nodule. Okay, improper muscle balance during phonation and chronic occurrence leads to scarring and nodule formation in the vocal cord. It is known as singer's nodule. Very common. কি করা হবে? Treatment? কোন প্রেসার বাড়লে এডিমা হয়? এক জোরে থেকে বল। কোয়াগুলেটিভ অসমোটিক প্রেসার, তাই তো? So that is incus edema. Laryngoscopy sacral formation. Okay, and laryngoscopy you have already seen. Okay, so these are the clinical anatomy. Okay, so it is another case where there is a cancer of the vocal cord. Okay, and it is due to human papilloma virus. So see, single nodule, it is not harming the person. Just there is one kidney, but there could be papilloma hote pare, right? And there would be cancer.
मैम मेरे को कैंसर और वॉइस चेंज हो
हाँ,
वॉइस चेंज हो
hoarseness of voice होते होते एक टाइम में
कितनी कैसे बोले